products and the drugs that have been used or are in trials for COVID-19. The drug that we're going to talk about today is the Favipiravir. Favipiravir has very recently been going into the phase 3 in India, and they are hoping that after the phase 3 goes successfully, they would be going out in public as well as phase 4 or in production. And the cost is really low, 103 rupees, which I believe is somewhere like $1.50 or even lesser. So that is a inexpensive drug, and it is going out in India. It is already approved for influenza in Japan. So it is an approved drug in some countries. There are trials for Favipiravir that are, of course, in India, Italy, UK, and USA. So let's talk about the Favipiravir. It's an interesting drug. We have talked about some of the medicines. For example, remdesivir is in a similar mechanism. So let's study. Good morning and welcome everyone. Okay, so here is the news. Glenmark's FabiFlu approved for coronavirus treatment in India cost rupees 103. So it's even, I believe, somewhere about $1. So meaning it is a inexpensive drug. However, medical experts cautioned against seeing it as a magic bullet to treat the deadly virus. And I would say the same thing right off the bat, that as much as it is promising, remdesivir doesn't have the similar effectivity as well. Just like this drug, it's not necessary that it is going to be this effective. So as it is used more and the phase 3 trial comes in, we'll know more. Anyways, this is one more hope for all of us. So here, Mumbai-based Glenmark Pharmaceuticals have launched a new antiviral drug, Favipiravir, with brand name FabiFlu, for the treatment of mild to moderate COVID-19 patients. The firm has become the first Indian company to commercially launch an antiviral drug. Indian drug regulator DCGI gave the approval for marketing and manufacturing. So that is the Favipiravir. I want to start from here. So this is another article. It is in Nature. So therapeutic options for the 2019 COVID-19 or novel coronavirus. In this here, if you see, in the virally targeting agents, the agents that are going to target the virus, approved nucleoside analogs, and I'll explain all of those things in a second. This is kind of a summary of what Favipiravir is about. Approved nucleoside analogs, Favipiravir and ribavirin, and experimental nucleoside analogs, that is remdesivir and galidesivir, may have potential against SARS-CoV or COVID-19. Nucleoside analog in the form of adenine or guanine derivatives target the RNA-dependent RNA polymerase and block viral RNA synthesis in a broad spectrum of RNA viruses. So this is the Favipiravir. Favipiravir is called T705, a guanine analog. So here the only correction I'll make, and I'll show you some other articles as well. It is not just a guanine analog, it is a purine analog, which means both guanine and adenine. Analog approved for influenza treatment can effectively inhibit the RNA-dependent RNA polymerase. So this is that enzyme that helps the virus replicate. So we'll talk about that too. So this is one. Then if you see here, this is a study. So look at the date for this study. This is May 15, 2020, May 15, so recent. Favipiravir strikes the SARS-CoV-2 at its Achilles heel, the RNA polymerase. So what they have done is, this is actually a study here, and what they're saying is a promising class of RdRp inhibitors or RNA-dependent RNA polymerase inhibitors are nucleoside analogs, and that is what we're talking about. Small molecule drugs that are metabolized intracellularly into their active ribonucleoside 5-triphosphates, we'll talk about that too, forms and incorporated into the nascent viral RNA by error-prone viral RdRp. So what we're saying is that we'll have nucleotides or nucleosides or bases that will become part of the virus RNA, and in becoming part of the RNA, it kind of disrupts that RNA or mutates it or causes issues with that RNA, that the enzyme that work on that RNA cannot work correctly afterwards. So that is a basic mechanism. And here, this is their study's result. And please keep in mind, this is an in vitro study. That is, it is a study done in a petri dish instead of a human being. So what they did was they took cells, viro cells, and infected them with SARS-CoV-2, and they presented Favipiravir 500 micrometer of T705, which is Favipiravir, and performed deep sequencing of viral RNA. So let's look at the results that they have. Keep these following things in your mind. Number one, they saw a 12-fold increase in G to A and C to U transition mutations. So we'll talk about what these mutations are. Then T705 or Favipiravir is acting as a mutagen. So that is their first action that they're saying. It acted as a mutagen. It caused mutation in the RNA. Capable of escaping the CoV repair machinery. The second thing that they observed was that the SARS-CoV-2 virus has a repair enzyme which proofreads the RNA that is being made. And if that RNA is damaged somewhere, it can correct that. And the Favipiravir can escape that correction mechanism. And actually, any antiviral that is going to work on SARS-CoV-2 at the RNA level will have to escape the proofreading mechanism. Otherwise, it would just be scooped out by the proofreader, removed from the system or the RNA, and it will not be effective. Then here, interestingly, the SARS-CoV-2 genome has an already low cytosine content, and I'll explain what that means. Treatment with Favipiravir, therefore, places additional pressure on the already unbalanced CoV nucleotide content. This is the basic study that they've done, and the results were beautiful, that they were able to see that the virus replication reduced, virus mutations occurred, and even virus RNA became damaged as well or terminated earlier as well. So here is another experimental treatment with Favipiravir for COVID-19, an open label control study. So this is an open label study. That means doctor knew that what they are prescribing, patient also knew what they are getting. So it is not a blind double blind study. This was March 18, and this is in China, I believe. Yes. So look at their results. Patients with laboratory confirming COVID-19 who received oral FPV. So this is another difference from the other drugs, that Favipiravir can be given by mouth instead of needing to be injected. So people can actually take this at home. So there's a comment from Jim that it is blurry. Hopefully it is just the network on one end and not everyone having an issue with the screen. So look at this, their results when they gave Favipiravir. So this is an in vivo study. Changes in chest computed tomography, that is CT scan, viral clearance, and drug safety were compared between the two groups. One was control and one was the group that was getting Favipiravir. For the 35 patients enrolled in the FPV arm and the 45 patients in the control arm, all baseline characteristics were comparable. So the patients were similar. A shorter viral clearance time, that was the first outcome. That the patients who were taking Favipiravir, their virus clearance time was short. So they recovered a little earlier. Secondly, the Favipiravir arm also showed significant improvement in chest imaging. So their chest lesions or the pneumonia or damage that was occurring to the chest had significant improvement. So the improvement rate was 91.43% in the patients receiving Favipiravir compared to 62.2% who were on the controlled arm. And then finally, in addition, fewer adverse events were found in the Favipiravir arm than in the control arm. So the Favipiravir treated patients had fewer adverse outcomes. In this open label before-after control study, FPV showed better therapeutic response on COVID-19 in terms of disease progression and viral clearance. With that, here is the Fujifilm, and this is March 31. They announced the state of a phase 3 clinical trial for Favipiravir. And please remember that Favipiravir is a an approved drug in Japan for influenza. So with this, let's look at the drug itself. So here we are. So let's start looking at this diagram from top here. So what happens is, this is the coronavirus. When it binds to our ACE2 receptor with its spike protein, it kind of fuses with our cell membrane. The RNA in the virus, or the genome of the virus, it appears inside our cell, uncoating of the virus, correct? So the RNA of the virus is in our cell now. Our cell have ribosomes, which are tiny enzymes. There are tiny machineries that will pick up this RNA, which is coming from the virus, and then they would convert that into proteins. So this is called the process of translation, that you take the genetic code and convert that into proteins. So the proteins that are made are here. So there are two big proteins that are made. These are called polyproteins, so PP1AB and PP1A. So these are two large chunks that are made. And if you see here, what I've done is I've made these tiny blue things in there. So this big protein contains small active proteins in it that need to be taken out, or that need to be removed from it. Just like a child's toy. For example, children sometimes go get the zoo or other templates, and they have a plastic sheet, and on that sheet there are stickers that are for zoo animals, and you just peel those stickers off. That is the same thing here. There is a bigger protein. In that protein, there are smaller proteins embedded in it, and we need to cut these proteins out. So these bigger proteins, called polypeptides or polyproteins, they also have active enzymes in them, which are called proteinases. Proteinases are the enzymes that would cut protein. Wherever you hear the word ases at the end of an enzyme, that means it does some kind of cutting. So proteinases are the ones that would cut a protein. If it was a DNases or DNase, it would cut a DNA. So here these viruses have their own proteinases. They cut proteins for each other, and the result is that these blue little proteins become free. At the same time, when this translation is occurring, what would happen is that we will also have one more protein called replicase. Replicase from the word replication, that would help in replicating the virus RNA. So this whole structure finally gives rise to more proteins after those tiny proteins are cut out. And here is a big complex that is formed. So this complex is actually virus protein complex. If you see here, there is a replicase in it, this red one. Then there is a transcriptase here, which would do some transcription, and we'll see that in a second what that is. And there is this big green one, which is the RNA-dependent RNA polymerase. This is that enzyme that is targeted by what is this drug that we're talking about? Favipiravir. This enzyme complex also have a proof reader with it. And this is what is important, that when the Favipiravir comes in and kind of causes issue with the RdRp and causes the RdRp to make incorrect RNA, that RNA will then be proofread by the proofreading enzyme, and that enzyme usually is successful in removing incorrectly built RNA. However, Favipiravir would escape this enzyme and would end up making or allowing the system to make bad RNAs. So this is the target. These two guys are the target. RdRp is the target and the proofreader is the target. Proofreader is the target in a sense that we have to escape it. RdRp is the target that we have to disrupt it. Now, what is the function of this guy, RdRp, or the replication transcription complex? This enzyme complex here, it takes the virus genome that came into our body. Remember, that was this genome. It takes this genome, converts this into a negative sense. The genome that comes in is a positive sense genome. That is just that it starts from the 5' end. It converts that into a negative sense genome, or you can say it is replicated. This replication is then fed into the same enzyme again, and then it would make more RNAs from it. The end result of all of that is that those RNAs that are made, they will go back into our ribosomes. Our ribosomes will now incorrectly, accidentally, unknowingly, will make proteins that are virus proteins. At the same time, there is virus genome that is being produced as well. So now virus proteins and the virus genome, when they will be collected together, will make new viruses. Good so far? Hopefully this makes sense. So this is an important area. Now what is happening here is, if I just give one more a few statements here, this positive sense RNA is converted to negative sense RNA, and for that we need RNA building blocks. And Favipiravir acts as a fake building block. So accidentally, this transcriptase complex will use Favipiravir as a building block and incorporate that into the RNA strand. That is how damage is going to occur, or mutation is going to occur. There is one more thing here, and that is Favipiravir can actually disrupt the RNA-dependent RNA polymerase as well. It is possible that that enzyme stops working altogether when too many, actually two, of the Favipiravir become part of the RNA. So that is an interesting one. This means it can cause premature RNA termination, or it can cause mutations. We'll look at that in a second. So hopefully, this much of the mechanism is clear, that virus came into our body, brought its RNA, we made virus enzymes unknowingly, accidentally. Those enzymes then took virus's own RNA and created more RNA for the virus, which will make virus proteins. And finally, that would help make virus. But in this process, lots of RNA is being made. And that is where this drug is going to target. So now let's see, how does it do that targeting? First of all, Favipiravir is a drug that when it is given and it goes in our cells, it does not disrupt our RNA, or it does not disrupt the production of our RNA. Instead, it only works on the viral or non-mammalian cells. So mammalian cells are safe from the activity of Favipiravir. That is why it would not cause cellular RNA or genomic damage to us. So that is one. Secondly, once it goes into our cells, over there it is activated, and activated to ribofuranosyl triphosphate, or FRTP. This is similar to another nucleotide bases, and we'll talk about that in a second. Finally, there is an enzyme in our cells that is called HDRPT. That enzyme helps with the activation of the drug. So the drug is actually a pro-drug. It needs to get into our cell and become activated, and this is the enzyme that helps. It is one of the enzymes that takes part in the activation. So now let's continue here. Look at this area. What happens is that when we are making our DNA or RNA, we use nucleic bases, or we use bases with phosphates, and we make nucleotides. These nucleotides have these bases in them, and bases are purines and pyrimidines. And here in the purines, we have adenine and guanine. This is the area, the ATP, that is this adenine A from there. And similarly, some of you would know GTP as well, which is guanine triphosphate. So these have these bases in them, adenine and guanine. Similarly, there are pyrimidines, which are cytosine, uracil, and thiamine. Thiamine is not present in RNA manufacturing. Thiamine takes part in DNA formation, while uracil takes part in the RNA formation. So these are the bases. The other important thing to keep in mind is that adenine combines with uracil. So what happens is one purine combines with one pyrimidine when we are making RNAs or DNAs. So adenine combines with uracil, and guanine combines with cytosine. So that is the base combination or base pairing. So see here, let's look at how this drug is actually going to cause issues with the RNA production for the virus. Imagine that this is the virus RNA. When the virus is now trying to copy its genome or its genetic material, what it does is it makes a copy of that here, and for every adenosine, there will be a uracil produced. This is why we are going from positive sense, for example, towards negative sense. So the opposing base is produced. That means that this produced thread, this copied thread, or I should say transcribed thread, is not yet sufficient, because we need to once again convert it back to this original form. So we'll have to transcribe it once more. So the first we do a replication where we make a copy, then we do a transcription where we make a messenger RNA. So here, this was the original. Wherever there was A or adenine, we would place while forming the RNA, we'll place a uracil. So adenine, guanine, for example, will have uracil, cytosine, because uracil combines with adenine and cytosine combines with guanine. And so I have the rest of the thread over here. You can see A and U, G and C, and so they are combined with the way they should be paired. Then what we'll do is that once this is formed, we can once again process another RNA from it, which will end up creating this original RNA. So this is how the virus will amplify the production of its RNA. Now let's see how Favipiravir is going to work. Favipiravir is an analog of adenine and guanine both, or adenosine and guanine both. So that means wherever there is A in the original thread, instead of that A, the RNA can incorporate an F or Favipiravir. So check this out. A, G. So for all of them, it is actually possible that we end up with Favipiravir, because it is an analog to A and G both. But what I did was I kept some preserved and I replaced some to kind of show you how the mutations will occur. So keep an eye on these A's and G's. So instead of them, here we have Favipiravir or a drug incorporated. Here is the normal base or nucleotide. This is normal. Drug incorporated, normal, drug, normal, drug, normal. Good? Now think about it, what's going to happen. When we make a RNA from this guy, in front of the F, remember F or the favipiravir here is going to act like an adenine or guanine. That means it's going to confuse the RdRp because adenine combines with uracil and guanine combines with cytosine. But if you put favipiravir, that can act like guanine or adenine. So how would the RdRp know what to place, C or U? It doesn't know it because favipiravir can act like both of them. The result will be that we caused a confusion for the enzyme. So on the other side here, if you see, for an A, we needed a U. So that is correct. There is an F and there is a U. For an A, we needed a U, and that is correct. For a G, we needed a C. That is also correct. And if you see here, for an A, once again, we needed a U, and there is this change. So if you see here, this thread, RNA thread, and this new thread that is produced because of the favipiravir's presence, they are significantly different. Look at it, U, C, U, C, U. Here we have U, C, U. We wanted a C over here, but instead we have a U here. Why do we have a U here? Because it is the favipiravir over here that can act as G or A both. So the enzyme doesn't know that I should put a C here or I should put a U here. This is a point mutation that we are getting. And there is a drug again over here and there is U here. So if we keep seeing C, that is correct. U, that is correct. Again over here, there should be a C here. But instead, because there was favipiravir sitting in there, the opposing side of that, or the pairing side of that, the enzyme is confused once more and it just put a U there. Instead, it should have been a C. So there is one more mutation you're seeing. And then this is correct, this is correct, this is correct, and there is one more mutation. This is mutation. Actually, this is correct, this is a mutation, this is correct, and then this is a correct one as well. So you see that there are a bunch of mutations. Now, let's look at a concept that is really important, and that is when favipiravir is present, it causes the nuclear bases to become varied in their ratios. So look here, this one is going to be very interesting. Look, it is a G or guanine. We know it combines with cytosine. But instead of the G, we have added a favipiravir here. And so instead of that, it is now a U over here. When this strand of RNA will be further transcribed, instead of U, we might get what? So U combines with G, correct? Sorry, U combines with A. So now here, we wanted a G to appear, but this U is going to give rise to an A over here when this transcription happens once more. This is called a G to A switching. We were supposed to have a G here, but instead of that, we will end up having an A over here. So that is a mutation. This is a G to A switching. Similarly, if you see here, we have a cytosine. Here we have a cytosine, and the opposing base is G. When we have an opposing F producer, which is the favipiravir, then it is possible that we have C over here, we have favipiravir over here, and when this F will be further transcribed, this might end up, instead of a C, it might end up being an A or being a U. So here we have a C to U mismatch, and that is one more kind of anomaly. The result of that is, if you see here, what would favipiravir do? It would do a couple of things here, or three things here. Number one, it can cause chain termination. What does that mean? What that means is that this was a transcription complex which has RdRp enzyme in it, and this enzyme has taken up the genomic RNA, and it is making a negative sense RNA from it. It is making a copy. Whenever two favipiravirs, these two F for example, whenever two favipiravir molecules are sitting next to each other in RNA, that would cause the RdRp to stop functioning altogether. It becomes so unstable that it just stops. So we call it a premature chain termination. That RNA is only replicated up to here, and further replication is stopped because there were two favipiravir that got incorporated in the RNA, and RdRp cannot handle it anymore. So that is one kind of damage to the virus RNA this drug would do, and that is premature termination of RNA. The other damage is that it can slow down the RdRp process. Because favipiravir is now sitting in there, instead of a guanine or an adenine, the molecular electrical charges are such that the RdRp cannot function that efficiently anymore. So favipiravir would stall the RdRp or the RNA replication and transcription processes. So the second thing is viral replication will reduce. Third thing is it is a mutagenic. Mutagenic means this. Remember here, instead of a G, we ended up with an A. That is a mutation. So favipiravir would cause single nucleotide mutations, which means here, instead of a G, we have a F, which would then translate into an A. So that is a single nucleotide mutation. Or it could have multiple nucleotides back-to-back mutated. And multiple can only be two, because if there are more than two, then the RdRp cannot actually function. It breaks at 2. So the total mutations it can produce in a consecutive order are either 1 mutation or 2 mutations. If there is a 1 mutation or 1 nucleotide mutation, that may be harmful or may not be harmful for the virus. But if there are two mutations, then that would cause the chain termination and stoppage of the viral replication. So this is the drug, Favipiravir. I know that we were talking a little bit of genetics, and that has an interesting outcome, and compressing it within the right time may be a little confusing, but I hope this was clear. So this is how Favipiravir works. In India, they are starting the phase 3 trial. It is inexpensive as well. There are trials that have started in other countries as well and have been started since March. So it's not a new news, but it is becoming more newsworthy because they are focusing on it in India. And I'm appreciative of the India's approach at this time. They took care of hydroxychloroquine usage in a very decent way. I think now 77 million people over there. And by the way, I'm not from India, so don't say that because I'm an Indian, that is why I'm praising India. I'm not from India. However, 77 million people have received hydroxychloroquine, and they have shown good results. So India has been doing some interesting things that are worth seeing for COVID-19. I had promised that I'll talk about India and its approach. Somebody reminded me last week as well that, hey, you did not talk about it. We will do that talk hopefully this week. So this is the discussion for today. I hope you all had a great weekend. I had a great weekend. Let's meet each other tomorrow. What would you like to talk tomorrow? So let me know what would you like to talk tomorrow, and we'll go from there. Please put that in the comments, and please like, subscribe, and share. Something interesting is that we are also banned on BitChute. So it's not that they are removing our videos, but we are not showing up in their search. So if I can just show you quickly what is happening. If I go here and I go to BitChute, although I am putting everything that is being removed from YouTube, I'm putting there, plus every new video that we are doing nowadays, I'm putting that over there. So if I look for Drbeen, there are only two results. One is the norepinephrine, that was the last video, and this is somebody who took my video and just uploaded it as their own. So this is also my video actually over here, but this is what we have. Fortunately, because you can find one of the videos from there, you can find the remaining, but you cannot find Drbeen channel over there. I think they have shadow banned us. So I do not know why, but that is what happened. So with this, thank you very much for joining. Stay safe and healthy. Please like, subscribe, and share. And I would see you tomorrow. Please let me know what would you like to talk about tomorrow. Bye-bye.